Good evening students. In this video we are going to discuss all diuretics possible along with their mechanism of action, right? If I miss any diuretic, you can always mention that diuretic in the comment section, but on my part I'll try to cover all the diuretics possible, right? So let's first of all draw a nephron, right? Our PCT, then our loop of Henle, right? Loop of Henle and our thick ascending limb of loop of Henle, right? Our DCT, right? Our collecting duct. Right. Now, let's discuss the mechanisms of diuretics one by one. Right. Let's start with the most famous diuretic, our furosemide. It's also called loop diuretic. Right. If we draw a small cell of PCT over here, on this side you have got sodium potassium pump, which is pumping sodium out and potassium in. Right, this leads to less sodium inside, right? And this sodium gradient is used by which ion channel? 1 sodium, 1 potassium, 2 chloride, right? 1 sodium, 1 potassium and 2 chloride, right? And which drug inhibits this channel, right? Our drug furosemide. That any drugs, furosemide, torsemide, right? They will inhibit this channel. Right? So what will happen if furosemide inhibits this? This sodium won't get reabsorbed, right? If this sodium is not reabsorbed, right, it will flow outside, right? Wherever sodium goes, the water follows, right? So water will also accompany it, right? If sodium and water are excreted out, this will lead to diuresis, right? One very important thing is that even potassium will be lost along with it. So furosemide, the one major side effect is hypokalemia, right? So in chronic furosemide therapy, you always have to give potassium supplementation, right? As per DCT is concerned, right, which ion channel is over here? We have got our sodium chloride symporter, right? Sodium chloride symporter. And it is also using the same gradient, right? We have got one cell of DCT and on basolateral side you will be having the same sodium potassium ATPase pump, right, which will be pumping sodium out and pumping potassium in, right, which creates a gradient and sodium and chloride gets transported. Now which diuretic inhibit this channel? Right, are thiazide diuretics, right? Most famous of which is hydrochlorothiazide, right? Your hydrochlorothiazide is going to right. Other drug are thiazide-like diuretics like chlorthalidone, right? It's a very popular drug given to antihypertensives, chlorthalidone. And indapamide. Right? Now, what other diuretics which are important, right? On this side, we have got are principal cells. In principal cells, right, we have got ion channels. What are those channels? ENaC channels. Very important ion channels, ENaC channels. And on this side, what we have got are sodium potassium ATPase pump. Sodium is pumping, so this is pumping sodium outside, potassium inside, which leads to less sodium inside, and ENaC is using this gradient to pump sodium inside, right? And this creates a negative charge on the inside of the cell, which pumps the potassium outside, right? So this negative charge sucks the potassium outside, right? So we have got some diuretics acting on this ion channel, right? So there are some diuretics which act directly on this ENaC channel, and that diuretic is amiloride, right? Amiloride. And triametrine. Right? So amiloride and triametrine, the diuretics, they directly inhibit your ENaC channels, right? And sodium potassium ATPase pump, right? There are some diuretics which inhibit the sodium potassium ATPase pump, and those diuretics are spironolactone, right? And eplerenone. These are also called potassium sparing diuretics, right? Now it's very important to understand that how these diuretics differ from furosemide or thiazide. Like in furosemide, the sodium is getting lost, right? Along with that, potassium is getting lost. So it will obviously lead to hypokalemia. But in this case, the sodium chloride symporter, the sodium is getting lost, right? And this sodium is very precious for body. So body will try to absorb this sodium, right? If body tries to absorb this sodium, it has to excrete potassium outside, and this will also lead to hypokalemia. So furosemide will also lead to hypokalemia, and thiazide, like hydrochlorothiazide, will also lead to hypokalemia. But in this case, the sodium won't be reabsorbed. So there will be no negative charge over here to suck out potassium, right? Since the sodium is not getting reabsorbed, the potassium won't come out. So this will lead to preservation of potassium. So both amiloride and spironolactone are called potassium sparing diuretics. They are actually mild diuretics, right? And given when we actually want to preserve potassium, right? And then apart from this, you know, there is it's a very important hormone, aldosterone, right? Now aldosterone is actually So aldosterone is actually stimulating all the steps, right? It is stimulating all the steps of it stimulates your ENaC channels, it stimulates your sodium potassium ATPase pump, right? So if we give a drug which can inhibit the synthesis of aldosterone itself, right? And that drug is your baxdrostat, right? If something can inhibit the synthesis of aldosterone itself, it will be a wonderful diuretic, and that drug is your baxdrostat. It is going to inhibit the aldosterone synthesis, right? Apart from that, there is also one very important diuretic which inhibits the action of antidiuretic hormone, right? If you draw a cell over here, collecting duct, right? Your ADH will come over here, act on which receptor? It will act on V2 receptor, right? How V2 receptor will act? It will convert your ATP to cyclic AMP. The cyclic AMP will increase the protein kinase, and the protein kinase will in turn recruit which channel? Your aquaporin 2 channel, right? AQP2, which is going to reabsorb water, right? So if somehow you block the action of ADH, right, so cyclic AMP level will fall, protein kinase level will fall, and aquaporin 2 won't be recruited. So water won't be reabsorbed, water will be excreted out. But in this case, the secretion of water will be, what we would say, water diuresis will happen, right? If you know, there are two kinds of diuresis, osmotic diuresis and water diuresis. In furosemide, your lot of sodium is going out, which is followed by water. In hydrochlorothiazide, a lot of sodium is going out, which is being followed by water, right? Even in amiloride, the sodium is going out, which is being followed with water. But in this, you will get lot of hypotonic urine, right? Because this is not due to excretion of sodium. This is because of lack of reabsorption of water through the aquaporin 2 channels. So which drug actually block the action of ADH, right? The drugs which block the action of ADH, right, are conivaptan, and tolvaptan. Right? Conivaptan and tolvaptan are going to block the action of ADH on V2 receptors, right? Now, apart from that, right, there are two very important diuretic drugs acting on PCT, right? If you recall from our previous video, right, in PCT, let's draw a cell of PCT, right? What lies on the basolateral side of our PCT? Our hero, sodium potassium ATPase pump, right? So this means less sodium inside, right? If there's less sodium inside is used by sodium, many gradients, many substrates are absorbed using the gradient of sodium, like phosphates, like sugar, like amino acids, right? So here we are dealing with sugar. So sodium, and along with that the glucose gets absorbed, right? Sodium is getting absorbed, and along with that glucose is getting absorbed, and what we call this transport channel as SGLT2, that is predominant in PCT. We have some quantity of SGLT1 also, but predominant form is SGLT2, right? Now which drug is inhibiting the SGLT2? Your dapagliflozin and canagliflozin. This is mainly used in diabetics to control diabetes, right? Because if glucose is not absorbed, the glucose will go out, but it's also a strong diuretic, right? So dapagliflozin and canagliflozin are going to inhibit your SGLT2 inhibitors. Right? One very important diuretic which actually acts on your PCT is acetazolamide, right? Now how acetazolamide acts, right? On this side, of course, you have got sodium potassium ATPase pump pumping sodium outside, right? Potassium inside. So this creates less sodium inside, right? If you got less sodium inside, then you have got one ion channel which will pump sodium inside, right? But H plus outside, right? Sodium, hydrogen and antipode. This is very important, antipode, right? Now But how exactly this H plus is being generated? You have got H2O plus CO2. It is being converted to H2CO3, which is being dissociated into H plus bicarbonate, HCO3 minus and H plus, right? H2O plus CO2 combining to form H2CO3, HCO3 minus and H plus. This H plus will go inside the PCT, right? And this H plus will combine with the bicarbonate coming through the filtrate, right? It will combine with H plus, H2CO3, right? H2CO3 will further break into H2O and CO2, which will be reabsorbed, right? So this whole cycle, this bicarbonate is being reabsorbed through this whole cycle. It is combining with H plus, forming H2CO3, H2O plus CO2, which is being reabsorbed. Now this H2O plus CO2, right, is combining to form H2CO3 and H plus is going outside, right? Now these two steps, conversion of H2O plus CO2 into H2CO3 and breakdown of H2CO3 into H2O plus CO2, both these steps are being catalyzed by one important enzyme called carbonic anhydrase, right? Now suppose if we block this carbonic anhydrase, then what is going to happen? If we block this carbonic anhydrase, right? So H2O plus CO2 won't combine to form H2CO3, right? And HCO3 minus, this H2CO3 won't break down into bicarbonate and H plus ion, right? If this H plus ion is not going out, then this sodium also won't come this side. If this H plus is not going inside the tubule, this Na plus won't get reabsorbed. And if this Na plus won't get reabsorbed, this Na plus will go into the tubule, and if the Na plus goes into the tubule, the water is also going to follow. If water follows, ultimately what you will get? You will get diuresis, right? So what happens over here, right? You have blocked the carbonic anhydrase, so H2CO3 won't get formed, your bicarbonate and H plus ion won't get formed, the H plus won't enter the tubule, so even sodium ion won't get reabsorbed. If sodium is not reabsorbed, the sodium will go inside the tubule and the water will also follow. This will ultimately lead to a diuresis. Now what are the diuretics which actually act, right, and inhibit this carbonic anhydrase? One very important diuretic is your acetazolamide, right? Acetazolamide is going to inhibit your carbonic anhydrase. Right? Now there is also one very important, a research level diuretic, right? It's not widely available, but yes. You know macula densa, what macula densa releases? Macula densa releases your adenosine, right? The same compound which causes vasoconstriction, right, in renal vascular bed, but vasodilation everywhere, right? Now it's a very important drug in a sense, very important compound in a sense that it also helps in understanding the mechanism of how exactly the tea makes you feel fresh, right? So all that, this caffeine, theophylline, right, they are basically adenosine receptor blocker, right? So they block the binding of adenosine to its receptor. With the help of adenosine, the brain senses that the body is getting tired. But if the adenosine receptor gets blocked, right, body won't know, the brain won't know that the body is tired, right? So similarly, what adenosine is doing, the macula densa is releasing adenosine, and what adenosine does, adenosine causes vasoconstriction of your afferent arteriole. If it causes vasoconstriction of afferent arteriole, it reduces GFR, and it also increases the activity of this sodium hydrogen exchanger. If it increases the activity of sodium hydrogen exchanger, more sodium will get reabsorbed, the water will also follow, so you will have get less filtrate, right? So both ways it is reducing the amount of urine, right? It is by the process of vasoconstriction of the afferent arteriole, it is reducing your GFR, and whatever filtrate is reaching, it is also increasing the reabsorption of that filtrate, right? So if we somehow block the action of adenosine, right, then what is going to happen? The adenosine won't act. If adenosine won't act, your arterioles are going to relax. If the arterioles are going to relax, you will have more GFR. If this sodium hydrogen exchanger will get inhibited because adenosine was actually stimulating it. If the sodium hydrogen exchanger gets inhibited, the more sodium will stay inside your tubules. If more sodium stays inside your tubules, the water is going to follow, right? And it will ultimately lead to diuresis, right? So name of that drug is rolofilin, right? Right? So what adenosine does? Let's write adenosine. What adenosine does? Adenosine causes vasoconstriction. You have got your glomerular capillaries. It will cause vasoconstriction of these capillaries. And by causing vasoconstriction, what it will do? It will reduce your GFR, right? And what adenosine further does? Adenosine increases the activity of your sodium hydrogen exchanger, right? So both these acts actually reduce the quantity of urine, right? Now, what drug we are talking about? We are talking about rolofilin, right? So if we write adenosine over here, so rolofilin is going to inhibit the action of adenosine. If it inhibits the action of adenosine, right? So what is going to happen? The vasodilation will happen. Which will increase your GFR. Then the activity of sodium hydrogen exchanger will go down. Right? And this will lead to diuresis because water is going to follow sodium everywhere. Right? So these are all the diuretics. If you say mechanism wise, right, it presents a complete picture. Just in case I have missed any diuretic, you can always write in comments, right? So let's sum it up, right? The furosemide, it is going to act on your 1 sodium, 1 potassium, 2 chloride channel, and this will lead to diuresis because it will block the reabsorption of sodium and water is going to follow, right? Your hydrochlorothiazide, it is going to block sodium chloride symporter. So sodium won't be reabsorbed, right? Sodium will be excreted out and the water is going to follow, right? In P cells, you have got your ENaC channels and sodium potassium pump. Amyloride and trimetrin directly blocks the ENaC channels, and spironolactone and aplanon blocks your sodium potassium ATPase pump, right? These two drugs, your amyloride and spironolactone, are potassium sparing diuretics, but the furosemide and hydrochlorothiazide lead to loss of sodium. So any chronic therapy of furosemide and hydrochlorothiazide, you have to supplement with potassium, right? One very intelligent drug is your losartan, right? So losartan leads to hyperkalemia, but hydrochlorothiazide leads to hypokalemia. So we combine the losartan and hydrochlorothiazide, right? So amiloride will directly block the ENaC channels and spironolactone will block your sodium potassium ATPase pump. There is one drug which blocks the aldosterone synthesis itself, and that is your baxdrostat, right? Now, one very important drug which blocks your SGLT2 receptors, right, and can cause diuresis are your dapagliflozin and canagliflozin, often used in diabetics, right? And this thing, your acetazolamide. The acetazolamide, by blocking the carbonic anhydrase, will block the chain itself, right? So H plus won't be excreted out and the sodium won't be reabsorbed. If sodium stays over here, it will be excreted out and the water is going to follow, leading to diuresis, right? Your adenosine is causing vasoconstriction, reducing your GFR, is also stimulating your sodium hydrogen exchanger. So if we block the adenosine from binding to its receptor, it will lead to vasodilation, increase your GFR, the activity of the sodium hydrogen exchanger pump will go down, right? Sodium will stay inside and it will be excreted out, water is going to follow, which will ultimately lead to diuresis, right? So hopefully this video has helped you in getting the overall picture of diuretics and their mechanism of action, right? Wish you all the very best. Thank you.